All right, we're good to go.
All right, thank you. Let me get my screen shared. All right, that looks good. So thanks again for joining. So I'll be talking about the NCTN and NCORP data archive, which is a repository of data sets from clinical trials, fairly recent clinical trials funded by the National Cancer Institute. So the goals for today are for you to be familiar with the data archive data sets. So what are they, what are in them, and how they can be used for your research. And then to also learn about the process for accessing the data and then publishing manuscripts based on those data. So I'll start with an explanation of what NCTN and NCORP are, if you're not familiar, just to get an idea of what sorts of data sets that you'll actually be able to find. I'll talk about the structure of the data sets that you can find. What are the variables? How are they organized, and so forth. I'll explain how to find data sets and see what data sets and variables are available. And then finally, I'll go over the process of actually requesting the data and using the data sets. So that's setting up an account with the data archive, getting this through their approval process, and like a DUA signed, a data use agreement signed by OSU. So the data archive, this NCTN NCORP data archive, is maintained by two organizations within the NCI. So within the NCI, you have the National Clinical Trials Network, the NCTN, and NCORP, the NCI's Community Oncology Research Program. So this data archive is maintained by these two organizations, and really it's meant to be a repository of data sets that are from trials that are funded or run through these programs. So the data archive itself is it's a central controlled access database, meaning that it's central in that you don't have to go request permission from the individual study teams or investigators or sponsors or anything like that. Everything, like if you want to request multiple data sets, everything happens through the data archive as a unit with the same process. And it's controlled access. So you do have to, you can't just get to a link on the website and download everything. You have to register an account, say who you are, get a DUA set up, and just have a very brief research plan. But other than that, you don't need to apply for any sort of competitive, any sort of competitive process or scientific review. So this database contains, again, data sets generated from clinical trials of either the NCTN or NCORP. And it's a fairly recent establishment, so it only generally includes data sets from primary publications of phase 3 trials that are published as of 2015. Or from non-primary publications of those same phase 3 trials published as of April 2018. So these being these phase 3 trials, right, are the ones that are the large-scale definitive confirmatory trials. You're not going to get data from the trials that are establishing dosage in phase 1 or the preliminary efficacy and safety studies. What they mean by data sets from publications is that the data set should be sufficient to broadly reproduce any results that are included in those publications. So but again, it's not just meant for being able to verify the results in the publications. You are allowed and encouraged, of course, to use this data for your own original analyses. There may, if you're looking to actually reproduce the analyses in the publications, there might be some minor variations just due to de-identification they had to do, or some data cleaning that was done after the original publication. But you should get broadly the same results. So the data sets are coming from trials by the NCTN or NCORP. So I'll talk about what those organizations are exactly and what kind of trials they run. The NCTN first, this network is made up of over 2,200 sites, predominantly in the US and Canada. It provides infrastructure for NCI-funded treatment and primary advanced imaging trials. So these are the trials that you're looking at to set standards of care, work towards FDA approval, testing entirely new treatments, validating biomarkers. And I'll say that if it's a trial from the US or probably even Canada that's focused on primary cancer treatment, the NCTN is probably involved. So you might not see, like if you're looking at a present publication of a trial, you might not see NCTN referenced directly, but you'll likely see that a trial is being supported by one or more of its member organizations, which are Alliance, ECOG, Energy Oncology, SWOG, Children's Oncology Group, and the Canadian Cancer Trials Group as well. On the other hand, NCORP is NCI's community oncology research program. They design and conduct clinical trials in cancer prevention, screening, supportive care and symptom management, surveillance, health-related quality of life, and cancer care delivery. It's made up of 46 community sites, 14 of which are considered minority or underserved community sites, which are the red triangles on this map. NCORP also has 7 research bases, which are basically coordinating centers for multi-center trials. Those research bases are similar to the NCTN organizations. They're Alliance, Children's, ECOG, Energy Oncology, SWOG, University of Rochester, and Wake Forest. So what's actually included in these, didn't go forward. So what's actually included in these data sets, the data archive entries are not actually by a single data set. They're entries by a trial. So each trial gets one entry, and that trial entry may have several data sets associated with it. The data sets are patient level. They're de-identified and have values for all variables used in the published analyses. So the de-identified patient level data is in a really nice sweet spot. So you can perform these detailed analyses and basically reproduce the analyses in the publications, or work with the same granularity of data as what they used. But on the other hand, since it's de-identified, it's actually not considered human subjects research. So you can get an IRB, it is IRB exempt, and you can get an IRB exemption letter fairly easily from Buck IRB. I'll say that some trials have associated imaging data available through the Cancer Image Archive. It's not actually hosted on the trials data archive, or on this data archive, but it will tell you and link to the Cancer Image Archive, where you can get the data separately through them. So there are 2 ways that you can use the data archive search engine to find data sets. And I'll use a trial whose data sets I've requested as an example here. So if you already have a specific trial in mind, you can find the associated data sets by providing one of the clinical trials identifiers. So there's the national clinical trial number, which is a number that would generally start with NCT and then a bunch of numeric digits. This is probably the most reliable one just because every publication should include it unambiguously. And you can easily recognize it by the NCT prefix. You can also use the study ID if it has one. So these are often given in like presentations and titles because they're a lot shorter, but it can be hard to figure out exactly like if an identifier is the correct study ID. So this trial is S1202 with the S standing for SWOG. But it's not like, if you have it, great, but it's not always as apparent that that's what they're talking about. You could theoretically use the full trial title. So for example, like just the entire long trial title that'll show up on the protocol. But again, it can be tricky exactly to figure out which title to use and how to enter it. And finally, you can use the PubMed ID of the publication associated with the data set. So if you found a publication, just plug in the PubMed ID. It's safest to use the primary publication, but just because it doesn't show up doesn't necessarily mean that the data set isn't available, especially if the data set is coming from a secondary publication. You can also browse, like if you don't have a specific trial in mind and you just are looking for it, like all trials in a disease area, you can use, you can browse by disease type just to see all the studies that are listed or that are available. So once you find a trial, you can view information about all of the data sets associated with that trial. So most data sets have most trials have multiple data sets associated with them. And how the data sets are exactly arranged might vary, but I think this particular trial is a good representation of what you will find in other studies, though it doesn't have any imaging data associated with it. So you have the subject data set, this first one, which has the patient level data, 1 row per patient, including eligibility, valuability. And so like, were they on the study long enough to like actually have any of the evaluations, treatment, demographic, and baseline information. So it's basically that central subject data set that you'll link all the other data sets to. You have the safety data set, which is 1 row per patient per adverse event. And then the assessment data set, which includes 1 row per patient per planned assessment time. So that's things like the primary outcomes and any biomarkers or intermediate outcomes, but generally not the adverse events. If you click through to each data set, you can view like if I were to click on one of these links, you can view the data dictionary to see what exactly you'll be getting from a request. So like what types of variables and things, not necessarily like a sample size count or any summaries, but you can of course find those in the original publications. So a data dictionary describes every variable in the data table. So that includes the short variable name here, the variable type, like is it a character, date, numeric, that sort of thing, a longer description of each variable. No, also I said date, but probably the most you'll get is like a year of. You probably won't get any dates more granular than a year, just for de-identification purposes. If you're looking at survival data or something like that, it all should be standardized to like, not dates, but like days or months or years survived after diagnosis or after the start of treatment. So you'll have a longer description of each variable telling you what it is. sometimes, well, for many variables, you'll have like the codes and code definitions for each level that the variable can take. So if it shows up in the data set as Y and N, you know that's yes, no, but it might not be so obvious, like these eligibility or ineligibility indicators or reasons are abbreviations for different reasons that a patient might have been randomized, but then later found to be ineligible for the study. Hopefully there aren't a lot of them. So this subjects data set included the alternative patient identifier. So it's not something like, it's specific to this trial, and it's often, it'll usually be sequential. It's just to help you link observations from different data sets all to the same patient, but then you can't link that ID to anything external from this data set, like an MRN or medical research number or anything like that. It has eligibility and evaluability information, whether and why they deviated from protocol, random assignment, baseline and demographic information, and then this one has some satisfaction survey responses for this trial. This is an example of the outcomes data dictionary. So it'll have that same alternative patient identifier that allows you to link between data sets, data collection time point, and then the values of observations that are collected at that time point. In this trial, it was pain and quality of life questionnaire scores. And finally, the adverse event data sets is simpler. It's just the patient ID, the adverse event code, the long description of that code, and then the highest grade of each adverse event experienced by each patient. So although you can see like what the variables are for any of these data sets, again, you can't see the sample size or summary statistics. You'd have to get that from the primary publication, or whatever publication that the data were reported in. So the process for requesting data is actually pretty straightforward. As long as really the biggest hurdle was getting the people on the OSU side to just sign the data use agreement. So first, you'll set up an account on the data archive site with your institutional email. So that's your OSU or osumc.edu email address. Fill out an online request form, just listing the trial numbers that you're requesting data from. And you'll have to submit a brief research plan, which is actually just like a one paragraph, almost like a paragraph from a specific aims page. I'll show you an example in the next slide. It's just for administrative review. It's not like competitive or going up for scientific review or anything. And once you plug in those trial numbers and the brief research plan, it'll automatically generate a data use agreement document for you and the appropriate office at OSU to sign. So it's actually non-negotiable. So there's no like back and forth negotiation involved with it, and you don't have to really write anything for it or propose anything. You're just saying, this is basically this is the purpose of the project. These are the data sets that I'm requesting. Everything else is template. It took me about a month to get DUA signatures from the OSU side. Part of that was just, I'll talk a little bit about it later, but it was just, they were a little bit confused on how to actually fill something out. And then, I guess we're swamped or just slow to respond. But once that data request was signed, I was able to get the data like within a couple days get it approved and download the data within a couple days. So at least from the NCI side, it's pretty quick. So This is an example of the brief research plan. It really is meant to be brief. It's not even like a page. I just had one paragraph, and it gets pasted directly into the DUA. The archive doesn't really give guidance about what should be included, but I just sort of stated the overall goal and gave them a kind of specific aim, which I've highlighted in red. So our overall goal is to determine how adverse events relate to patient perceptions of the effect of duloxetine for treating aromatase inhibitor associated musculoskeletal symptoms. We will identify the effect of duloxetine on patient perceived benefit in patient subgroups defined by the post-randomization adverse events and reductions in pains using principal stratification. And then just beyond that, a little bit more statistical detail, but it's not, you don't have to have the research plan does not have to include any sort of data analysis plan, like we're going to use these models and these methods, or this is the sample size you need. So there's not really that sort of, you don't, although it's certainly good practice, you don't need to, it's good practice to write sort of a protocol outlining what you plan to do. It's not part of the review process of the NCI here. If you've written NIH grant applications, I just thought about it like stating again, state a specific aim with an overall goal pasted in the front of the paragraph. So the data uses agreement is pretty simple from your perspective. You fill out the first parts of the request form, and then you get the unsigned DUA to download. You send this DUA to the appropriate office internally at OSU. So for OSU, it's the Technology Commercialization Office, but I'm not sure if, I don't know if the Med Center has their own version of this. To be honest, I just worked through the OSU office. So again, the DUA is non-negotiable. The TCO didn't have any problem with that, except that they ended up using the wrong email address at first. NCI, at least at this point, wanted NCI needs an email address that goes to a specific individual. So like the name.number address of somebody at the TCO. TCO originally wanted to use the contracts at osu.edu, which is where you go to, like, you can contact them there to start the process of getting the DUA set up. But when they actually sign it, they have to use the email address of an individual. I've been told in chat that it's the same for the Med Center. You also go to the OSU TCO. So you do have to list everyone that will have access to the data. So later additions have to be made through a DUA amendment approved by the NCI. So it's not just like, if you have a study that you're doing with OSU data and you need to add somebody, often you can just have a quick form submission in Buck IRB to add them to the IRB protocol and everything, and it goes through fairly quickly. I haven't done it. I haven't had to add anybody to the DUA later, but I imagine it could be a little bit more cumbersome just because you again have to get these people from two different organizations to sign off on it. But it's not forbidden. It's not something that's going to be declined. You just have to give it some time, I suppose. So there are some important but fairly easy stipulations in the DUA, mostly relating to the publications. So before submitting for publications, you have to send the manuscript to the NCI to review, and they may bring in what they call NCI collaborators involved in the trial. So that's going to be somebody like the trial sponsor for a drug trial or something like that. you have to include this boilerplate acknowledgement of the data archive and specific trials in your publications, just saying like, this work used this data set from the NCI archive, and it was derived from this trial. Once the manuscript is published, you have to send its PubMed ID within 60 days, but they'll send you a reminder. I think they just scan PubMed for papers with that acknowledgement and send out a reminder when it shows up. And then finally, the DUA expires after 3 years. So after that, you either have to renew the DUA or destroy any of the individual data that you hold. So just for more information on like getting this started and requesting bird support. So this is the link to the data archive. This is where I was pulling those screenshots of just browsing through the search engine and the trial entries and the data dictionaries and all that. If you want to request bird support on a paper, you can go to the myccts.osu.edu or submit a ticket to the Center for Biostatistics. So again, this goes through pretty quickly, but and thanks again for joining. And I'll leave this slide up a little bit so you can get a screenshot or whatever you need. But also, I'm happy to answer any questions about either the data archive or just the process of doing this.
Thank you, Patrick. That was really informative because I did not know about this data archive. So thank you for walking us through it. And so, yeah, we have some time for questions. And if people are formulating questions, and I'll look at the chat too if anyone's posted in chat, but you're welcome to kind of come on and ask it. I had a question because I was kind of curious about just using this data and like some examples of how they can be used. When I think of trials, especially like phase 3 trials, I think of it, they're very focused. There's a primary outcome, and the goal is to really demonstrate efficacy along this primary outcome. And some, there are secondary outcomes and whatnot, but it's different from, for instance, like a cohort study where the idea is just, you collect a bunch of information, you can come up with spawn off new ideas and projects. Trial, at least in my mind. And I'm just curious, like, how are folks using this to garner new information? How are people pulling data from multiple trials? I mean, certainly meta-analysis is one thing I can think of off the top of my head, but I'm sure there's more creative things that people are doing. And I'm just curious what have you done with them?
Sure. So the meta-analysis is one obvious way to do this or to use this. And it's particularly useful just because it's often hard to get the individual level data for meta-analyses. So this is really useful in being able to get those more detailed data sets. It's another popular use seems to be, let's say you have these variables that were from the original trial, but they didn't necessarily look at or report like analyses of different associations, or maybe you didn't, I guess, depending on whether it's statistical or clinical interest, you might be interested in something like a subgroup analysis or a different way of modeling the data. So an example, I think it seems like subgroup analyses are particularly popular, but I would suggest being cautious with that. One, first, because these, unless the trial was run, probably these trials don't have the power, like the sample size necessary to do really in-depth or reliable subgroup analyses. Just because if they had subgroup analyses, like if they had designed the trial to be large enough to support subgroup analyses, they probably did it because they wanted to do subgroup analyses themselves. You're probably not going to find enormous trials with the data that that can support a lot of subgroup analyses that haven't already been done. The second, just remember that when you're doing this sort of subgroup analyses that unplanned, you do kind of put yourself at risk for having issues with like data dredging and that sort of thing. Just you can keep looking until you find something, but it doesn't mean that you should or that it will be a reliable result. I'll give you an example of what I was doing. So this is the primary publication of the trial that I was giving as an example. And so it was a trial evaluating duloxetine, which is often used an antidepressant, but also can be used for pain management, as sort of a supportive medication to prevent some significant adverse events related to hormone therapy for breast cancer. So originally it was looking at like what impact does duloxetine have on pain was a primary endpoint, and they had a secondary endpoint of what it was a health related quality of life. I'm trying to find. It was a specific measure, of course, but it was sort of a function. I think a functional quality of life secondary endpoint. And I'll show here. Here's an example of where you find that clinical trial number, or of course the DOI down here. So they went through this analysis of, this is the apparent impact that duloxetine has on quality of life, on pain, but then they also sort of had this let's say very secondary or even tertiary endpoint of like how they asked at the end of the trial, how satisfied, like a satisfaction question, do you feel that overall this whatever treatment that you received, they were still blind at this point, do you feel that whatever treatment you received was beneficial or was worth it? And they'd say something like, it was beneficial and it didn't have serious adverse event or didn't have significant adverse events. It was beneficial, like even though it had adverse events, it was still beneficial on balance, or it wasn't worth it because of these adverse events. So what I did was take this analysis and see, or take this data set and say, what's the association between what actually happened to them when they were on the trial and what they ended up saying in their satisfaction survey. So was it like, if you had adverse events, was that contributing to you saying that the treatment was not beneficial, or like maybe as long as there was some substantial level of improvement of pain, people were willing to put up with most adverse events just because they considered them less bad than the pain that was being treated. So you can look at these. I like to think of it as if your primary publication is saying this is the effect of the treatment, then a lot of secondary, a really fruitful question for secondary data analysis is, well, why? Like, how did that exactly come about what was sort of the the mechanism, at least in at the level of variables that we have available. What kind of associations can we see? This trial didn't really have biomarkers, but if you do find a trial that has a lot of biomarker data, that can be something that you could look into. So that sort of mediation or moderation analysis, or potential for surrogate endpoints or biomarker evaluation, things like that.
So actually, I'll pick it back on that, and then real quick, because maybe other people, folks want to ask questions, is the biomarker. That's a, I'm curious, so do trials have biomarkers, and do they also have like genome-wide data available for patients in these trials, for some of these trials? And if they do have that, is it something that's available through this data archive, or is it linked to some other data archive where that, genomic data is stored?
So I'd say you should be able to find at least limited biomarker data here. I say limited just because, if they collected it but didn't report it in any of their publications, it's not necessarily going to be included here. So it's just something that they collected for exploratory data analysis and just haven't gotten to it yet, then it probably won't be included. The genomic data, I think, is iffier. So you could certainly have like individual mutations if they were considered like a biomarker in the trial, but actual like full sequencing data, I would be surprised if let's say, I haven't seen them say either way, but I would be surprised if it was in there, just because of how big and how identifiable it could be in theory. These data are, I want to say they're very de-identified. Like there's very little like personal information in there, just like age, sex, race, that sort of thing. There are, I haven't done this myself, but there are, of course, like repositories for this genomic sequencing data, and I have seen statements from the NCI or the NIH more broadly that they are willing to help you link data sets link together data sets from different NIH or NCI sources that aren't in like that are about the same people. But I think my impression is that tends to be more of a one-off project, and I imagine would take much longer and require different considerate, like it would probably be more strict as well, just because it does have more potential for identifying people.
Thank you. Good answer. Other questions? I don't see any questions in the chat. Okay, well, if there are no other questions, thanks again, Patrick, for this presentation, because I thought it was pretty interesting, and I didn't know anything about this beforehand. So thank you again, and appreciate everyone coming, and hopefully see you at the next month's talk. Have a great day, everybody.
Thank you.